So I wanted to just talk to you a little bit today about what we do at Regeneron and kind of explain how drug development works for an antibody company. That's what we make. We make monoclonal antibodies. And why genetics is really important for that. So at Regeneron, our goals for human genetics are primarily around three things. So first and foremost, there's target discovery. This is the idea that if we understand some of those associations between a genotype and a phenotype, if we know what a region of the genome or a variant or a gene, what its impact is on biology and health, then we can make an antibody that can either mimic the effect of that particular variant or it can ameliorate the effect of that kind of variant, which then allows us to take someone's disease state and make it better, basically. So make a drug out of a genetic insight. But once you have a drug or once you have a drug target, there's a couple other things you can do with genetic data as well. So indication discovery is really about taking a drug that you already have or taking a drug that's currently in development and understanding if it can have another impact. So a great example is something like aspirin that's good for a headache, but also some people may take it at low doses for stroke or for trying to help with heart disease. So the idea of once you have something that you know can pull some biological lever and can impact someone's health in a positive way, maybe there's some other things you can do with that. And this again is something that genetics can help incredibly well with. And we've already seen a lot of examples of the way the association analyses can tease apart how different phenotypes or how different parameters around a person's health and even their socioeconomic status or ability to get a good job can really be teased apart from these genetic signals. And so we do the same thing here in drug development in trying to understand if there are other ways we can deploy drugs. So another thing we can do is take a drug and try to understand how it affects people who take it, and if there are effects of the drug that are different for people who have different genetics. And so there's a few examples of this that people may be aware of. Things like blood thinners are well known to have different impact on some people who carry certain variants. And so of course that's important to understand if we want to be making drugs that are useful for our patients that are not causing harm. Then we want to know if there are ways of genetically classifying individuals, if somebody is a carrier of a given variant and then maybe they're not going to respond to that drug, so we wouldn't recommend giving it to them, or maybe they'll respond really well so they'll get a different dose. Now, at the end of the day, Regeneron as a company historically has been about mouse genetics. So we are a company that has made enormous numbers of transgenic mice. And the idea of using mice as a model for human disease is common across a lot of companies. We were hugely committed to it, and then there was this realization that by partnering with people, by really committing to large-scale genome sequencing, to really understanding the genetics of humans within the human, that can actually really feedback loop in a very effective way with our mouse program. So we can look for targets, we can look for insights into the human genetics that we can then model in the mouse, and we can see things in the mouse that we can then look for signals in the human. So we now, with this large-scale genetics, this ability to sequence genomes and exomes very rapidly and somewhat cost-effectively, we can now really have a more virtuous and a more rapid cycle of development around human genetics and mouse genetics. Now, at Regeneron, the way that we've implemented large-scale sequencing is really we are full-on committed. So I would argue that there is maybe no other effort anywhere in the world that is sequencing as many people as we are, as fast as we are. And the way we did this was by taking our in-house expertise in automation, which was developed out of our efforts to automate a lot of the things we were doing in mouse genetics, and bring them to human genetics. So we have an automated biobank. It's maybe not as impressive as the biobank you all have, but I like to call it a vending machine for samples. You just push a number, you get a sample out, and those can then be immediately brought over to our library prep automation. So our sequencing and lab operations group, led by John Overton, has built an enormous amount of automation to really enable this ability to sequence people at very large scale, as rapidly as possible. And that's important because we are interested not just in the common disease variants, we're not just interested in the polygenic diseases like American spelling, but we're also interested in rare diseases. And if you're interested in rare diseases, you need to have very large numbers, and the best way to get to that insight rapidly is to sequence very rapidly. And I'm not going to say much about this. We've already talked a little bit about it in discussions around the importance of informatics for this kind of data. I will say, since this is actually what I do on a day-to-day basis as the head of Genome Informatics, we are 100% cloud-based shop. Everything that we do is in the cloud. Everything that we do is essentially has to be scalable. And so the idea of buying local computers, the idea of having a bunch of physical hardware in the building just doesn't work for us. And I think you know what you see generally is non-local compute is transforming the world in a variety of ways, but it's definitely transformative in genetics, where not only do you need this lab with this amazing automation that allows you to sequence all these samples so rapidly, but you have to be able to analyze them too. So I just want to talk a little bit about the way that we think about genetics, what kind of the four main buckets of projects for ours are, and point out a little bit some of the partnerships that we have. So at this point, the RGC has over 40 collaborators. This represents over 1 million samples. And these are primarily with academic groups, because the academic groups are historically the groups that have done the kind of sample collection and the kind of phenotyping that you all have done with the UK Biobank. And so as an entity that wants to really get at insights about the genetics and share that with the world, we need to go to the people who both have experience in analyzing that kind of data, the enthusiasm for sharing the data and sharing it with the world, and then, of course, somebody who actually has samples. So these 4 buckets for projects for us is general population projects. I'm not going to describe those. I think everyone here knows well this model, because the UK Biobank is maybe, if not the single best example of this kind of project in the world. It is certainly one of very few projects that operates so effectively at such a large scale, where there is such great phenotype data and so many individuals. We also do a very large number of these family studies, these very rare disease, what a geneticist would call Mendelian disease or highly penetrant disease, where we look for individuals who cluster in families with very serious genetic issues, and then we sequence as many of the individuals in the family as we can to try to understand what those genes cause. These are incredibly useful for understanding those monogenic diseases, but because these diseases are so rare, they're not impacting large numbers of people, and they often will not give you much insight beyond kind of where you started. So if you know you're looking at a particular disease type, you might gain insight into that disease, but it's not always more broad. Founder populations are somewhat interesting. This is a case, it's sort of like a general population, but it's where the individuals within the population have a much broader genetic sharing. So great examples are like the Faroe Islands or Iceland, where a relatively smaller number of people founded a population, and everyone, or more or less everyone within that population, shares that variation. From a genetics point of view, and particularly for us, it's very interesting, because while these kinds of populations will carry fewer rare variants than you would see in a population that wasn't a founder population of the same size, for the rare variants that they have, that they will be enriched. So it gives you an opportunity to try to identify a lot of individuals who are carriers of rare variants, that then lets you leverage that data to understand better how that relates to phenotype. And then in this last bucket are these phenotype-specific cohorts, and these are often really, once we have a genetic insight, we want to get deeper into it. So we'll identify people, again, primarily academics, who have collected very large, or somewhat modest sometimes, because some of these diseases can be somewhat rare, cohorts of individuals where we can understand better. But the point of this is all of this feeds back into each other. So the insights we gain from family studies, we can use that to try to marry that data with general population studies. Founder populations and phenotype-specific cohorts can come in as well. And so ultimately, genetics is not just about taking these individual studies and analyzing them on their individual merits. We've already heard a little bit about replication and how important that can be. It's really about bringing as much as possible the data together, because fundamentally, all genetic data sets should fit together at the genetic level. We're sequencing people. So I just wanted to mention this. I'm sure everyone here is aware of this, since we've already heard some questions about it. But we have this great collaboration with UK Biobank and GSK, where we're really excited to sequence 500,000 participants over this 3 to 5 year period. This, I think, I don't want to say this is the largest project that anyone has ever envisioned, because I think there are other larger projects that people are talking about, but my opinion is this is the most likely project to actually succeed at this scale anytime soon, because you all have the samples, you have such great phenotype data, and you have such clear commitment. The people in this room absolutely show that to getting this done. And so within the first year, this year, we'll get 50,000 samples done, and then hopefully as soon as possible, we'll get all 500,000. I did just want to point out a little bit kind of the other end of what we do. So we're not just forming partnerships with people and bringing samples in and sequencing them and sharing them with people. We are fundamentally an academic research unit as the Genetics Center that is embedded in a pharma company. We are all about publishing and sharing our results. So this is a great series of examples. I'm not going to go into much detail. I will point out that each one of these projects is in collaboration with between 1 and 5 or 10. Actually, the one in the middle, it's collaboration amongst collaborations. So again, there's this theme of cross-collaboration in genetics. But for this paper, there was a big study, and we actually acted as the replication cohort. So people were able to look at our data and confirm that what they were seeing in these other studies that had come together to try to understand the lipid pathway. And just to walk through this, the point of this is we're looking at 3 different genes. So there's ANGPTL4 and ANGPTL3, which you can tell they're similar because they have the same name but a different number. And they relate to this lipoprotein lipase here. And the idea is that by looking at all of the genes in the pathway, we can understand how they interact. So decreasing activity here is decreasing catalyst, similarly in ANGPTL3, and then in LPL, increasing activity decreases and vice versa. And putting this all together then creates a holistic program where we can really understand the fundamental biology, and that is so important for trying to make a drug that is effective for people, that doesn't cause problems, and that is something that can really impact people's disease in a positive way. So I just wanted to end on this example. I do want to point out, since we've had a long discussion earlier about return of results, so we are a research sequencing only shop at Regeneron. By the American rules, what that means is the data that we make cannot reach patients without it having some separate assay done that actually is validated in a clinical way. And the idea is, before you tell a patient something, you have to have a level of certainty about it that's maybe a little bit different than the data that you would look at for research purpose. And I think people generally understand this because we've discussed this so much. But what we've done with the Geisinger healthcare system, where we've sequenced 100,000 patients, and they have exquisite detailed medical records going back to the 90s. They have chosen, similar to the strategy that was described earlier today around the imaging decisions, they have chosen a very small number of variants where they have clear evidence that these variants have impact. They do a clinical validation of them. You can see the list here. And I believe if you go to the Geisinger MyCode website, they talk a little more in detail if people are interested in this. But the idea is these things that so clearly are correct in terms of the genetics and where there's some action that can be taken. So there's this great story of Barbara Barnes, who had an unseen stage 1 fallopian tube cancer, and that was discovered because she was sequenced as part of this collaboration. They saw this variant in the BRCA1 gene, which has an association with increasing breast and ovarian cancer risk. I think many people are generally aware of this now. And so they did some follow-up with her and found this issue she had, but now she's doing incredibly well, and she has an expected great outcome. And so I think this is a great example of what can be achieved, but I have to tell you the discussion around the imaging stuff really resonates with me, because the idea of just giving people genetic data more broadly, I don't know that necessarily works. And as Geisinger has implemented, they're reporting in these relatively restricted things, which is, I think, quite common, at least in the US. Anyway, that's all I had. Thank you so much, and I guess let's move on to who's next.